Okay, in this video, we're going to look at lipids. Okay, we're going to divide this video into two classes of lipids, fats or triacylglycerides and terpenes. So this will be the first video. We'll focus on triacylglycerides. Basically, lipids are the fat-soluble portion of biological molecules. They're typically very hydrocarbon rich and usually do not contain amine or carboxylic acid groups, since these groups would be ionized at biological pH and thus they would tend to be more water soluble. Okay? All right, so fats come in a variety of classes. Triacylglycerides are classically what you think of as fat molecules, and other nonpolar organic molecules that also fall into the class of lipids include waxes, terpenes, prostaglandins, and steroids. And what we're going to find here is that these diverse structures actually have a common biosynthetic origin. This one molecule here called acetyl SCOA. All right, and we'll see the significance of that molecule as well. All right, so let's look at the first class here, triacylglycerides. All right, so triacylglycerides are examples of somewhat complex or fancy esters, okay? If you look at them, what you see here is three ester groups, okay? And since they're esters, they can be hydrolyzed. This is frequently done with sodium hydroxide. Okay, we'll talk about the significance of that in a second. All right, initially you get from that process sodium salts of carboxylic acids. Okay, and the alcohol part of the ester goes away as this molecule called glycerol. Okay, and when you acidify the products here to a pH below the pKa of your average carboxylic acid, you get these three carboxylic acid molecules, and we refer to these molecules here by this term fatty acids. They're the acids that are derived from fats. Okay, this process here, it actually has a name, this reaction, saponification. Okay. And this is how soap was made for many years. Okay, we talked a little bit about soap and detergents in the carboxylic acid chapter, but you take a naturally occurring fat and you hydrolyze it under basic conditions, and you generate a mixture of the sodium salts of the fatty acids and glycerol. This whole material all together here constitutes, I guess we'll just say, a very traditional type of soap called lye soap. Okay, and this has remarkable cleaning properties. Again, we talked about this in the carboxylic acid chapter, how when these fatty acids dissolve in water, they create regions of nonpolarity in the aqueous solution, so you get the ability to dissolve greasy things in water. Okay, so here's a ester hydrolysis in process. Anyone recognize this person here performing the ester hydrolysis? Well, it's a very famous person. This is Brad Pitt playing the character of Tyler Durden's in the movie Fight Club, and he's performing an ester hydrolysis on human fat that he pilfered from the liposuction clinic. Basically, the soap that he made this way was very high quality, and he sold it to high-end department stores for $20 a bar. Okay, so there's a movie Fight Club that kind of centers, or that has ester hydrolysis as one of its major themes. Okay, actually, I think if you saw the movie, you wouldn't Actually, that would not be the most memorable part of it, but anyway. All right, so let's look at some trivia about fatty acids. Okay, and these are these compounds that we see down here. Okay, so let's look at some of the facts about them. All right, so they typically contain linear chains, no branching, and they also contain an even number of carbons, including the COH group, and there's usually more than 10 carbons. We'll talk about why they have to have an even number of carbons when we look at their biosynthesis. They can be saturated. That means that the chain contains nothing but sp3 carbons, or they can be unsaturated. That means the chain would contain one or more alkene groups. Saturated fats are usually solids at room temperature. They're more commonly derived from animal sources, but one major source of fats, the palm family, actually produces saturated fat from a plant source. Okay, and the solid nature is due to the fact that they can stack on top of each other. We'll look at an energy minimum conformation of a fat in a future slide. Basically, when we draw in their lowest energy conformation, which is the all anti or the zigzag, where it's just straight across the paper, we'll see that the chains can stack on top of each other quite easily. Unsaturated fats are usually room temperature liquids. They're more prevalent from plant sources. When nature produces an alkene in a fat molecule, it's almost exclusively the cis alkene. Okay. However, the trans alkene is more stable. So the cis alkenes that you get from the plants, there are conditions upon which you can turn them into trans alkenes, and that's the famous trans fatty acid that you've heard so much about in nutrition science. And unsaturated fats don't tend to crystallize very readily because there's a kink in their chain that makes it very difficult to stack them on top of each other. Okay. The unsaturated fatty acids can be monounsaturated, which is a property associated primarily with olive oil, or they contain lots of alkene groups. Things like corn oil and soybean oil contain multiple alkene groups. They're polyunsaturated fats. Okay, so let's look at some common fatty acids. Up here we have the saturated ones. Okay, C12 is lauric acid. C14 is myristic acid. C16 is palmitic, stearic, and then arachidonic. Notice that they all contain even numbers of carbons, and they all are room temperature solids. Their melting point is above 25 degrees C. Okay, below here are some unsaturated fats. Okay, so we have this palmitoleic acid. Oleic acid derived from olives is the highest melting of them at 13 degrees. Linoleic acid and linolenic acid are polyunsaturated acids, and this last one, arachidonic acid, is also unsaturated. Notice how their melting points are all below 25 degrees C, and the highest melting point of them all have only one double bond. The more double bonds you get in the chain, the harder it is to stack them. Okay, so here's the energy minimized conformation of stearic acid and oleic acid. Notice that here, basically, we draw this. These are what we call space filling models. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Okay, so that's the most stable conformation we have for stearic acid. If we look at, if we wanted to draw the Newman projection like citing here, what we would see is that this carbon here is anti with respect to that carbon right there. And we call this the all anti or the zigzag conformation. All right, these molecules, saturated fats, will stack on top of each other very nicely. It doesn't matter how long the chain is. They some will just hang out on the end a little bit longer than others. Okay, but in oleic acid, the double bond being right here, this is our cis-alkene group, introduces a kink in the chain that makes it difficult to stack. And oleic acid only has one alkene group. Some of the other unsaturated fats have multiple alkene groups and exacerbate the problem. So for this reason, they have a hard time crystallizing. They don't find a nice ordered way in which they can pack, so it's harder for them to achieve the solid state. Okay, let's look at some fats and their natural sources. Okay, so first we have animal fats. These are derived from animals. What you see here is that we have saturated and unsaturated. These, in most cases, these numbers aren't going to add up to 100 because this is only part of the spectrum of fats that you can have. Okay, all right. So if we look at these animal-derived fats, what we find here is that there's a kind of a skewing that there's more of the saturated than the unsaturated, but there's actually quite a lot of the unsaturated. They contain both kinds of fats. Okay, butter, I guess, contains the lowest or the shortest chains, but all the others contain larger chains. It's important to remember that even though we associate saturated fats with animal-derived fats, most of these still have an appreciable amount of unsaturated fat in them. Okay, all right, so let's look at some others. Let's see, I think this is, in this column is oleic, linolenic, and okay, linoleic and linolenic. All right, so this is the coconut is derived, of course, from the palm family, and this contains almost all saturated fat. Olives contain virtually all of one kind of fatty acid chain, oleic acid, which is a monounsaturated fat, and that's what's associated with the heart healthiness of olive oil there. Palm oil, okay, you can see also has a lot of saturated fat. Peanut oil and corn oil have a lot of polyunsaturated fats, and that's actually they're not quite as heart healthy as the monounsaturated fats. Okay, so let's look at food science here, okay, or food chemistry. All right, so it's easier to get fats from plant sources than animal sources. Nobody has to die. I guess, well, your corn plant has to die sometimes, but so you can get these unsaturated fats quite readily, but how do you make these room temperature liquids behave like the animal fats? Okay, so butter is a solid and it's spreadable, but if we took like just olive oil and tried to spread it around, we'd find, well, you can't get it on the knife because it's a liquid at room temperature. So chemists discovered that all you got to do is just take the unsaturated fat and subject it to a hydrogenation reaction. Okay, all right. So if you subject it to lots of hydrogen and platinum, what you'll get is completely saturated fat. Okay, and if you've ever dealt with Crisco and things like this, I think these are mostly for the hydrogenation reaction that goes to completion. They're somewhat hard. They don't make good spreadable material. If you took completely saturated fat and tried to spread it on your bread, you'd tear up your bread. Okay, so about 50, maybe 80 years ago or so, they discovered that they could run the hydrogenation reaction, but not to completion, and turned vegetable oil into something that had the consistency of butter. Okay, and this is what we will call partially hydrogenated fat. Okay. All right. So in the process, though, these hydrogenation catalysts, they isomerize double bonds. They move them in position, and when the new double bond is created somewhere else, it can be created cis or trans, and trans is actually more stable. So it wasn't until people started hydrogenating their vegetable oils that you started getting a significant amount of trans fat in the diet. So this is somewhat rare in nature. It does occur in nature, but not to a very large extent. But if you look at partially hydrogenated vegetable oil, there's actually quite a significant amount of trans fat. Okay? All right, so this is a problem now that there's three types of fats: unsaturated, saturated, and trans fat now. So it's dietary science think that monounsaturated or unsaturated is better for you than saturated, but there's some caveats there. Monounsaturated, like olive oil, is better than polyunsaturated, like peanut oil, as far as heart healthiness goes. So what we have here, and then, so saturated fat is not as bad as trans fat. So what sometimes you'll see modern day margarine type or butter substitutes. A lot of times what they'll do is they'll just take vegetable oil and completely hydrogenated oil and mix them. Okay, and that tends to get rid of the trans fat. Okay, all right. There's another reason why people want to hydrogenate vegetable oil too, and that's the fact that we associate alkanes with chemical inertness, whereas alkenes do a lot more reactions. And one of the reactions that they can do is autooxidation. Okay, and what they'll do is they'll add a O2 to the allelic position. Okay, and this was going to give us a lipid hydroperoxide. And these are actually this is what we call rancidity. Okay, the fat doesn't taste as good after it's gone through this process. So in order to get potato chips to have really, really long shelf lives, you would want to have as much saturated fat as possible. So anyway, that was the other motive for doing that. That if there are no double bonds, you can't. The fat will not go rancid nearly as quickly. All right, let's see, just a couple of dietary trivia things. What's an omega-3 fatty acid? This means that the third carbon from the chain is an alkene. Okay, and linolenate esters derived mostly from fish. It's really the only really good source of the omega-3 fatty acid. Some other plants like walnuts and flaxseed, I think, also produce some quantity of omega-3, but not nearly as much as fish oil does. Okay. The other issue is, why is coconut oil considered heart healthy even though it's almost all saturated fat? And that's because the major fat contributor only has 12 carbons, and it's burned quicker, burned for quick energy rather than stored. So it's processed kind of like a carbohydrate. Okay, we're going to end this by looking at fatty acid biosynthesis. And if you recall the carbohydrate chapter, we looked at how glucose becomes this molecule, acetyl-SCOA. The key thing to note here is that this is a thioester. Okay, and this is the molecule SCOA drawn out. Okay, you can see it's kind of lengthy, so we don't want to draw all of this stuff right here. Okay. All right, we're going to use this molecule, NADH, nucleotide, adenine dihydrogen phosphate, something like that. All right, so this molecule is going to transfer one of these hydrides here to become aromatic, and this is a reducing agent, but it only reduces a C double bond O that's complex to an enzyme, so it's not nearly as reactive as sodium borohydride. For oxidations, we're going to do NAD. We're going to use NAD+, which is what you get when you remove the hydride from this molecule here. The motive for removing the hydride from NADH, of course, is that you make an aromatic molecule. And this can accept the hydride from molecules. So this does the reverse of reductions that NADH does, and it can only oxidize something that's activated through enzymic complexation. It's not nearly as good, or not as a universal oxidant like chromium 6 species or nitric acid or something like that. All right, so we're going to swap initially the SCOA for this molecule here, and what you'll notice here is that for the most part, SCOA and this are the same all the way to this point here. So the chain is really only far, far away from the acetyl group where there's any difference between the acyl carrier protein that's what ACP stands for, and the SCOA. All right, so we're going to swap the SCOA for the SACP. All right, sodium bicarbonate is going to give us the malonate system. Okay, so carbanions derived from esters and ketones have pKa's, the acids that give those have pKa's much higher than water, so they can't exist. So we don't really make anions by deprotonation. We make anions by elimination of carbon dioxide, okay? All right, so this reacts like the anion, okay? All right, so this is going to add to the carbonyl group, make this compound right here. All right, this is going to lose the SACP, and it's going to give us this compound here, the acetyl acetonato SACP. And if we look at this process going from here, here to here, this is just the Claisen condensation, okay? A reaction very familiar to us, okay? All right, we're going to reduce the ketone to the alcohol. We're going to dehydrate it. The enzymes sort of scrunch this molecule into such a way that it gives only the cis-alkene, during the dehydrogenation process. We're going to now transfer a hydride in a net hydrogenation. This gives us the butyryl CoA. And we're going to repeat steps 2 through 5. This has 4 carbons. This has 6 carbons. This has 8 carbons. So you can see now why you have an even number of carbons. We build them up by acetyl-SCOA 2 carbons at a time. So all of our fatty acids in nature end up having even numbers of carbons. Okay, every once in a while, you're going to not reduce the compound or not reduce the crotonyl SACP or any of the derivatives here where you have a double bond, and you're going to build the fatty acid from the unsaturated ketone, and that's where you get the unsaturated esters when that process happens. So we could have repeated steps 2 through 5 on this compound here, hypothetically, right, on the crotonyl SACP, and we would end up with a compound that has a double bond in it. That's how we get the unsaturated fats. Okay, we're going to finally just look at some molecules that are derived from fats. Okay, so waxes are esters of fatty acids with the corresponding alcohols derived from fatty acids, forming the alcohol part of the ester. These form the coatings of various fruits and things like that that protect the inside. So supposedly, I don't advise that you try this experiment, but you could take an orange and dissolve it in, or put it in a solution of cyanide, then take the orange out and wash the rind out, and the orange would be okay to eat. Again, don't try that. But the idea here is that you've protected the inside of the orange from the aqueous environment outside by this fact that it's coated with this very nonpolar wax-like substance. Okay, a very important type of fatty acid is a phospholipid, and here's an example here. Basically, we've taken one of the ester groups and replaced it with a phosphate ester. These phosphate esters are very important in the cell membrane systems. Okay, so cell walls are made of phospholipid type molecules. Okay, so here's a common acid, arachidonic acid, and it goes through a rather interesting series of biological transformations in the presence of oxygen into a prostaglandin. Prostaglandins affect a lot of properties like bronchial constriction, platelet aggregation, induction of labor, and they regulate inflammation and many other roles. Aspirin and ibuprofen are actually what we call prostaglandin synthetase inhibitors. They inhibit the conversion of arachidonic acid into prostaglandins, and that's how they function as anti-inflammatories. Okay, all right, so this is the end of the fat slide, and we'll look at another class of lipids in the next video, terpenes. Okay.